Welcome back, biology students. We are still in our genetics unit. We have discussed a lot of cellular processes so far this year and in this unit, like DNA replication. We've talked about mitosis and meiosis. We just finished protein synthesis. And we know that these are all very important processes for all living organisms. But what happens if a mistake is made during one of these processes? What implications might that have on an organism? In today's lesson, we're going to answer these questions. Now, we know that cells make mistakes. These mistakes can be simple substitution mistakes where the wrong nucleotide is substituted in a process maybe like protein synthesis. Or it could be a mistake made during DNA replication that affects the process of mitosis. There's different mistakes that can happen. We'll talk about a few in this lesson. And these mistakes can have a range of effects on an organism. Now, when they occur in DNA or RNA, these mistakes are known as mutations. So by definition, and you need to make sure you write this in your notes, a mutation is a change in an organism's nucleic acids. And there are two types of mutations. So we have gene mutations, which is what we're going to discuss. We're going to focus on that in today's lesson since we're in our genetics unit. And then when we get to heredity, which is our next unit, we're going to come back and talk about chromosomal mutations. So today's focus is on gene mutations. All right, so we have different types of gene mutations. You can have what's known as a point mutation, also known as a substitution. And in a substitution mutation, this sounds exactly what you think it would be. A mutation where one nucleotide is put in place of the correct nucleotide. Now, usually a mistake like this is caught and fixed by the enzyme DNA polymerase or RNA polymerase in RNA. But if not, the substitution could permanently change an organism's DNA. Now, notice it says may. We're going to talk about some examples of substitution mutations that have an effect on an organism and then some that do not. So we'll talk about that later. But an example of a mutation, a substitution mutation that does have a tremendous impact on an organism is sickle cell anemia. And if you don't know, sickle cell anemia is a blood disorder, and it's caused by a substitution mutation where a single nucleotide in the DNA sequence is substituted for another. And that substitution causes the substitution of an amino acid known as glutamic acid to be swapped for another, valine. And now this mistake results in the shape of a blood cell to be sickled. So blood is typically supposed to be spherical, but in sickle cell anemia, the individual has sickle-shaped blood cells. All right, so here's an example. Here at the top, you see the original DNA sequence. So we see T, A, C, T, G. This is the way it's supposed to be. But in a point mutation, a substitution mutation, we can see that one nucleotide is just simply swapped for another. So instead of thymine, we have cytosine. And this could impact the organism, or it may not impact the organism. Now, in a frameshift mutation, this is going to involve two options. We might have the insertion of a nucleotide, or we might have the deletion of a nucleotide. So when a nucleotide is added or taken out of a nucleotide sequence in a DNA sequence, this is going to shift the entire sequence. So we call this a frameshift mutation. So frameshift mutation is where we have an insertion or a deletion and the entire frame just sort of shifts around. So if you'll look on the left of your screen at the bottom, you can see an example of an insertion mutation. So here we have the original sequence, and we can see in an insertion example, we have cytosine, which is just kind of plugged in here between this nucleotide and this nucleotide. And so what that does is that's going to create a frame shift where the DNA sequence is going to be altered as a result. And because the tRNA is going to read these in codons, it just completely alters the reading frame. You can also have a deletion example. So here we can see we're supposed to have T, A, A, C, T, G, but in a deletion, the thymine is completely wiped out. And so again, this causes the entire nucleotide sequence to shift over, and that could create the wrong amino acids. All right, if I ask you to read this sentence, you shouldn't have any problem, right? The dog sat. Simple. However, if we were to take out one of the letters, this would create a frameshift situation, and all the letters would kind of shift over. Now, if I were to provide you with this sentence initially, it would be hard to recognize that this is supposed to be the dog sat. And the same thing happens in an insertion or a deletion or a substitution mutation. It causes everything to just kind of shift over, and it becomes sometimes difficult for the tRNA to read the correct amino acid. And so the deletion changes the reading frame, and this could result in codons that might code for different amino acids. All right, let's suppose we insert an extra letter. So we have our original sentence that's supposed to read the dog sat, but if we insert an extra letter and everything shifts over, again, you can see how difficult this would be to read the dog sat from this example. And so the insertion changes the reading frame, which again is going to result in codons that might code for different amino acids. All right, let's look at a substitution mutation where we are supposed to have AAG. And if you use your codon chart and look up AAG, you will see that AAG codes for the amino acid lysine. Now, lysine is, it assists in helping enzymes bind to their substrates, which is super important. So let's say we have a substitution mutation and we have the A substituted with a C. Okay, so instead of AAG, now we have CAG. And if we use our codon chart, we're going to find that this codes for the wrong amino acid. And so this substitution could affect protein folding. It could even destroy the protein's function, which would be a tremendous problem to the organism. We call these sort of substitution mutations missense mutations. So a DNA change that results in a different amino acid being used to build a protein at a specific position is called a missense mutation. All right, let's look at another substitution example. Let's say we're supposed to have UAC, which codes for the amino acid tyrosine. And we're going to replace that C, we're going to substitute that C with an A. Okay, now this also codes for the wrong amino acid. Here we're going to get a stop codon. So this substitution would result in what's known as a premature stop codon. And we call this a nonsense mutation. So in a nonsense mutation, a DNA sequence change is going to happen that causes a cell to stop making a protein before it's finished. All right, let's look at another example. So let's suppose we're supposed to have the codon AAG, which codes for lysine. And let's say that the G is substituted with an A. Okay, so we have AAA. And if you look at your codon chart, you'll notice that AAA also codes for lysine. So this is what we call a silent mutation. This is a change in the DNA sequence that has no effect on the amino acid. All right, now gene mutations can be caused by a variety of factors. So changes in the DNA sequence can occur due to errors during replication, during cell division, exposure to environmental mutagens like radiation or certain chemicals, or even spontaneous changes that happen just naturally in the cell. So essentially, any factor that alters the nucleotide sequence of a gene can lead to a mutation. Now, some mutations, well, let me go back. Mutations, you do need to know, they're not uncommon, but it is a misconception a lot of students think that all mutations are bad. When we hear the word mutation, we often think something negative. But it's very important for you to understand that not all mutations are bad. Some are. Some are very harmful, like cystic fibrosis. Cystic fibrosis affects the body's mucus and sweat glands, and so it can cause a variety of different difficulties like breathing problems and digestive issues. And that would be an example of a mutation, of course, that's harmful. Some mutations are helpful. So there is a mutation associated with immunity of typhoid fever, where certain variants can provide natural resistance against the disease. So that would be an example of a mutation that's actually helpful. Some mutations have no effect. So the mutation that causes red hair is a genetic variation in the MC1R gene that affects the production of melanin. And so this is not going to harm the person. This is not going to be helpful to the person. It has no effect. So again, mutations are not uncommon. Some are harmful, some are helpful, and then some have no effect. Now, usually organisms have many tools within the cells that help repair these mutations or find these mutations or fix them before they become a problem. We've talked about before DNA polymerase or RNA polymerase is the proofreader enzyme that usually does a really good job of proofreading and catching these mistakes. But occasionally mistakes do happen within the cell. We also have something called a mutagen, which could be responsible for gene mutations. So mutagens, examples of mutagens would be like UV rays from the sun. Some industrial chemicals are considered mutagens, different carcinogens. Infectious agents like viruses and bacteria are also considered mutagens, and these mutagens cause mutations because they can directly alter the DNA sequence through different mechanisms like base substitutions, insertions, and even deletions. All three of those we talked about today. All right, if you need to go back to any part of this lesson, make sure that you do that. Make sure that you took good notes, and I will see you in the next lesson.